Good day everyone, we are Group 3 and today we are presenting on Case 1, Iron Storage. We hope you enjoy our presentation.
Dietary iron has a functional role in the body in that it is an essential constituent of hemoglobin, which is involved in the transport of oxygen from the lungs to the tissues. It is also a component of myoglobin, which aids in the development of healthy connective tissues and supports muscle metabolism. Additionally, iron is critical for neurological development, growth, and cellular functioning. Figure 1 illustrates the recommended daily allowance of iron. It is notable that these values are affected by gender, age, chronic diseases, and menstruation.
Case Study 1 presents a 54-year-old male who has denied exposure to toxic chemicals and metal fumes. His complaints include weakness, lassitude, moderate weight loss over 7 months, and visibly darker skin over the past few years. His vitals recorded that his pulse, respiratory rates, ECG, and chest x-rays were all normal. However, his blood pressure was found to be high with a reading of 145/75 millimeters of mercury. Medical investigation showed that the liver was firm and moderately enlarged. The spleen was palpable, and a biopsy of the liver indicated that there was fatty vacuolization of hepatocytes and moderate deposits of hemosiderin in the cytoplasm of cells. The patient was diagnosed with hemochromatosis, a type of iron overload, which was confirmed by a differential deferoxamine test.
Green leafy vegetables, food from animal sources, particularly red meat, shellfish, and legumes are good dietary sources of iron. Figure 2 illustrates other iron-rich foods. As indicated in the case study, the 54-year-old man was diagnosed with hemochromatosis, which contributed to his iron overload. Since dietary iron is important for functions previously mentioned, the aim of his diet should be to reduce the dose of dietary iron to manageable levels, and this can also be achieved with phlebotomy or chelation therapy. Since heme iron is easier for the body to absorb than non-heme iron, he should consume a diet incorporating more plant-based sources of non-heme iron, unlike meat, poultry, fish, and seafood, which contains heme iron. Moreover, he should consume calcium supplements or calcium-rich foods like milk, yogurt, and broccoli, as calcium can inhibit the absorption of both heme and non-heme iron. Vitamin C supplements should also be avoided, as this will contribute to non-heme iron absorption.
As previously mentioned, iron has many different roles in the human body. We obtain iron from the foods that we eat. In the body, iron exists in two states, either the ferric state, which is the Fe3+ state, or it can exist in the ferrous state, which is the Fe2+ state. When we consume iron-rich foods, the iron from that meal is either heme iron or non-heme iron. We obtain heme iron from foods such as meat, poultry, and seafood. In this case, the iron is sourced directly from the animal's heme proteins, hemoglobin and myoglobin. The iron exists in the ferrous state and is easily absorbed by the body. Non-heme iron is sourced from plant foods such as vegetables, beans, legumes, nuts, and seeds. In this case, iron exists in the ferric state, which has a much lower solubility, and it is not easily absorbed. Therefore, before absorption can occur, the ferric iron must be converted to ferrous iron. In the small intestines, there are epithelial cells which line the duodenum, and these are called enterocytes. Located on the surface of these enterocytes, there are special ferroreductases named duodenal cytochrome B. This enzyme is responsible for reducing iron in the ferric state to the ferrous state. Once in the ferrous state, divalent metal transporter moves iron from the surface of enterocytes to the cytosol. At this point, iron has two fates. Ferrous iron can be oxidized back into ferric iron and stored as endogenous ferritin, or it can exit the enterocyte in the ferric state through exit proteins called ferroportin. It then enters the bloodstream to be transported to various tissues via transport protein transferrin. Regulation of iron absorption. In the body, there are no regulatory mechanisms of iron excretion. This means to prevent the body from total iron deficiency and iron overload, iron absorption must be carefully regulated. This is also known as iron homeostasis. The main regulator of iron absorption is a liver-synthesized peptide hormone known as hepcidin. Hepcidin binds to ferroportin, the exit protein we spoke about earlier, and this binding event causes ferroportin to disintegrate. As a result, iron is unable to exit the enterocytes, which leads to significantly lower levels of iron circulating in the plasma. Additionally, this encourages iron to be stored. In cases of iron deficiency, the liver produces lower levels of hepcidin, which promotes iron transport throughout the body. In addition to hepcidin, iron absorption can be inhibited and enhanced by certain foods. One example of an enhancer is vitamin C. The ascorbic acid chelates iron, which makes it more absorbable by the body.
Figure 3 shows iron absorption, transport, storage, and recycling. Following absorption in intestinal mucosa, ferrous iron enters the bloodstream via the ferroportin on the basolateral membrane. However, iron is circulated via the transferrin protein only in its ferric state. Therefore, the ferrous iron must undergo an oxidation reaction performed by Hephaestin or ceruloplasmin to be converted to ferric iron to enter the bloodstream. Ferric iron in the blood binds to an apotransferrin molecule to form transferrin, which facilitates transport to major sites for iron utilization in the body, such as the bone marrow, liver, and spleen. These sites possess transferrin receptors on their membranes to facilitate binding of the transferrin molecule. A receptor transferrin complex is formed after this binding, and it is endocytosed, allowing the complex to enter the cytoplasm. In the cytoplasm, iron is released for metabolism and storage, and therefore the receptor apotransferrin complex is formed. This complex then moves to the membrane for further separation to form apotransferrin, which is returned to the circulation, and the unbound transferrin receptor to be recycled. This slide shows the proteins involved in iron transport and storage.
Hepcidin can enter the general circulation and block the action of ferroportin, which is responsible for the release of iron into circulation. The primary objective of hepcidin is to decrease the plasma ferric iron concentration. Additionally, it plays a critical role on spleen macrophages. The spleen is composed of many macrophages. If a red blood cell is damaged or becomes old or weakened, it enters the spleen, where it is engulfed by splenic macrophages. These digest the damaged or old blood cells into smaller components, one of which is iron. Ferrous iron can be released into the general circulation, whereby it undergoes recycling in the bone marrow for erythropoiesis. Distinctively, hepcidin will inhibit the action of ferroportin, thus preventing the release of ferrous iron in circulation and subsequently lowering ferric iron concentrations. Hepcidin can also prevent ferrous iron absorption from the small intestine's lumen. These are the factors which regulates the concentration of ferric iron in the plasma, as well as leads to its storage. Excess iron in the body signals pathology. This may arise due to hemochromatosis. This is a disorder in iron metabolism characterized by excess iron absorption, saturation of iron-binding proteins like ferritin, and deposition of hemosiderin in the tissues or organs. Primary hemochromatosis or hereditary hemochromatosis occurs when an individual absorbs too much iron due to a genetic mutation in the HFE gene which regulates hepcidin. Acquired or secondary hemochromatosis is produced by excess of iron intake like dietary iron overload. In primary hemochromatosis, since the HFE protein is unable to associate with a transferrin receptor, this results in an increase in the expression of the divalent metal transporter known as DMT1. Excess DMT1 expression leads to an inappropriate increase in intestinal iron absorption. Additionally, the inability of the HFE gene to activate the HAM gene encoded in the hepcidin protein contributes to the decreased expression of hepcidin. This causes the unregulated presentation of ferroportin in the basolateral membranes of enterocytes and in the plasma membranes of macrophages. With no control over iron release to the blood from these two cell types, plasma iron concentration increases. Since there is reduced to no HFE-mediated regulation of the transferrin receptor, too much iron is absorbed into tissues, particularly the liver, pancreas, and skin, as hemosiderin. This leads to the progressive symptoms of hemochromatosis. Iron deposition in the liver leads to cirrhosis, and in the pancreas causes diabetes. The excess iron deposition leads to bronze pigmentation of the organs and skin. It is important to note that excess free iron in plasma stimulates Fenton chemistry to generate highly reactive oxygen species known as ROS and hydroxyl radicals. This is detrimental as it can damage other cellular components, the phospholipid membrane of mitochondria, lysosome, and DNA. Thus, tissues and organs that undergo high oxidative metabolism are more prone to damage by ROS and exhibit greater sensitivity to adverse consequences of iron overload, such as cell degeneration, cell death, and its advanced toxicities like cirrhosis. In hemochromatosis, the liver is usually the first organ to be affected. Initial symptoms include weakness, abdominal pain, change in skin color, and the onset of symptoms of diabetes mellitus. In advanced cases of hemochromatosis, there will be issues like congestive heart failure, testicular atrophy, as seen in the figure. Alcohol consumption, iron intake, and excessive vitamin C ingestion can also be the cause of the symptoms of iron overload. Treatment of hemochromatosis before there is permanent organ damage can restore life expectancy to normal. Treatment involves removal of the excess body iron, which is achieved by phlebotomy.
Phlebotomy is the removal of blood. When it is done as a treatment, it is defined as therapeutic phlebotomy and is recommended to patients who have high iron levels in their blood. After treatment with therapeutic phlebotomy, about 500 cc's of blood is removed and the iron level in the body drops by approximately 250 milligrams. There are 3 phases of therapeutic phlebotomy, the first being the induction phase, the second being the transition phase, and the last being the maintenance phase. The induction phase is where therapy is initiated and many factors are put into place such as diet, the patient attending scheduled phlebotomies, and continued education on change behavior. In the transition phase, the iron levels may be erratic. Phlebotomy may need to be twice a week, once a week, or once a month. The transition phase aims to transition the patient into the maintenance phase, and during this transition period, the iron levels fall steadily over a period of time. In the maintenance phase, the patient has achieved healthy iron levels and can maintain these levels by donating blood regularly over a period of time. The maintenance phase aims to ensure that the patient remains symptom-free and iron levels will be tested every 3 months. The length of time it takes for a patient to reach the maintenance phase varies. The body produces new blood to replace that which has been removed during treatment. The new blood then acts like a sponge, drawing iron out of the bodily tissue, which is stored in ferritin. This synthesizes hemoglobin in freshly produced blood. When iron forms in the new hemoglobin, it can be removed in the next phlebotomy treatment.
Thank you for your time. Are there any questions?